Hello everyone, today I am going to talk about a brief history of fungicides. We all know fungicides are important in plant disease management, especially for those plant diseases which are caused by fungi. So in this lecture, we will know how the fungicides have been developed over time. At present, we have hundreds of chemicals to address plant disease problems in many crops and against a varieties of fungal pathogens. Now, how these fungicides have been developed over time will be discussed briefly in this lecture. The word fungicide has been derived from Latin words fungus and cido. Cido means to kill. So, fungicide refers to any chemical that can kill fungi. In contrast to the term fungicide, we have another term called fungistat. Fungistat refers to the chemical which inhibits fungal growth temporarily. Fungistats do not kill fungi, they inhibit the growth temporarily. Similarly, we have terms like bactericide, bacteriostat. Bactericide kills bacteria. Bacteriostats inhibit the growth of bacteria. So we have these terms fungicide, fungistat, bactericide, and bacteriostat. We see the mention of use of sulfur, the earliest known substance which was used for plant disease management. Homer, the ancient writer, around 1000 BC, mentioned the therapeutic properties of sulfur on plant diseases. So, sulfur is the first thing which was used for plant disease management. Democritus, 470 BC, recommended controlling plant blights by sprinkling plants with the olive grounds left after extraction of the olive oil. So, it is a kind of use of botanicals that we know today or plant extracts in the management of plant diseases. In the 9th century, Shurpal's Braksh Ayurveda mentioned about tree wound dressing, tree fumigation, and seed treatment. So, this is one Indian literature that mentions about plant disease management. In 1637, Remnant reported seed treatment with sodium chloride for controlling stinking smut of wheat. In 1705, Homburg used mercury chloride as a wood preservative. So here you can see gradually substances are coming like sodium chloride, mercury chloride. These salts are coming into picture for plant disease management. In 1755, Tillett, who is known for his work on wheat, he reported seed treatment with lime or salt and lime for management of wheat diseases. In 1761, use of copper sulfate was reported for managing stinking smut of wheat. So perhaps this is the first use or report of use of copper sulfate. Later on, we will see how copper sulfate has been used extensively for plant disease management. In 19th century, we have some important developments as far as fungicides are concerned. In fact, the base of plant disease management, the chemical management of plant diseases started in 19th century, in the later part of 19th century. Prevost in the year 1807 from France showed effectiveness of copper sulfate in controlling stinking smut. So we have been seeing a series of attempts to manage stinking smut disease of wheat using various chemicals. In 1821, Robertson used sulfur to manage peach mildew. In 1833, Kendrick he proposed lime sulfur for management of grape mildew. Then finally, in 1882, the first discovery of a fungicide which has revolutionized the chemical management of plant disease is your Bordeaux mixture. So control of downy mildew of grapes with Bordeaux mixture was reported by Professor Millardet from Bordeaux University of France. We'll talk about this Bordeaux mixture, which is a copper fungicide, later in some other presentations. How this Bordeaux mixture is prepared, what was the concentration, what was the background of discovery of Bordeaux mixture. It was actually the observation made by Professor Millardet which led to the discovery of this fungicide. After Bordeaux mixture development, we have seen a series of other attempts to improve the performance of Bordeaux mixture. Bordeaux mixture is still a very good fungicide because it has multiple sites of inhibitions in the target fungus and it can control a wide range of fungi, even bacteria. Bordeaux mixture is basically a mixture of copper sulfate, lime and water. Here copper is the main fungitoxicant. Now copper is sometimes phytotoxic. To reduce its phytotoxicity, lime is used where we have calcium. In 1885, Ozane first tried copper sulfate in India. In 1887, Masson in France introduced Burgundy mixture. He replaced one component of Bordeaux mixture, that is lime. He replaced calcium carbonate of Bordeaux mixture with sodium carbonate and it was called as Burgundy mixture. Though Burgundy mixture is not used anymore, but historically it is important to know that there is one alternative to Bordeaux mixture. In 1888, Trillet reported fungicidal property of formaldehyde. And formaldehyde was used in the earlier times to manage soil-borne plant pathogens. We know formaldehyde is a fumigant. So when it is applied in soil, it can kill soil pathogens, soil-borne plant pathogens. In 1900, Selby used formaldehyde for soil treatment to control onion smut. However, formaldehyde is not used anymore for controlling soil-borne diseases because formaldehyde indiscriminately kills all microbes present in soil. And it is not good for soil health to kill all the microbes which are present in the soil. So use of formaldehyde for soil treatment is not recommended anymore. Now we come to 20th century. In 20th century we have some very good developments as far as fungicides are concerned. In 1904, Lawrence used Bordeaux mixture in India for the first time. Then sulfur was used for the first time in India. In 1913, Reim introduced organic mercurial for seed treatment. Burns in 1914 first used mercury chloride in India to treat seed potato. Then Hilson in 1925 used organomercurials for seed treatment for the first time to control sorghum smut in India. Now we know these mercury compounds, inorganic or organic, are banned in most of the countries, including India, because mercury is a heavy metal. It gets accumulated in the food chain, which in the long run is deleterious to health, human health, animal health, or it is not environment friendly. So use of mercury compounds as fungicide is banned. However, when they were found to be very good fungicide, they were used. In 1934, Tisdale and Williams first reported the fungi toxicity of dithiocarbamates. These are sulfur compounds. We will see more about dithiocarbamates in our subsequent presentations. In 1940, Cunningham and Charvellet introduced chloranil as seed protectant. For seed treatment, chloranil as a fungicide was used. Then zone of inhibition technique for laboratory bioassay of fungicide was given by Burlingham and Reddish. Now this technique is very important to test any fungicidal compound in the lab. There are many techniques to test that. We have poison food technique, zone of inhibition technique, or disc diffusion technique. We also have agar diffusion technique. So those techniques are very useful for testing fungicides in vitro. Then we have now some very important developments of fungicides. In 1943, Diamond et al. Introduced ethylene-based dithiocarbamates as fungicides. This is also one sulfur fungicide, organic sulfur fungicide. In 1952, one chemical, captan, was used as a fungicide. Kittleson, he introduced this fungicide, captan. So captan is also known as Kittleson's killer. It is named after the discoverer of the fungicide, captan. Then the landmark development happened in the year 1966 when systemic fungicides were discovered. So far, whatever fungicides we have talked about before 1966, those were all contact fungicides. Those fungicides, including captan, then sulfur fungicides, copper fungicides, Bordeaux mixture, those were all contact fungicides. They do not enter the plant system. They act where they are applied. So, in 1966, oxathiin, under which we have carboxyne and oxycarboxyne, two different compounds. These are systemic fungicides. So when they are applied, they enter the plant and get translocated inside the plant and kill the pathogen away from the point of application. So that benefit was achieved with systemic fungicides. Now these systemic fungicides were used to manage rust and smart diseases. Plantvax and Vitavax are two trade names of fungicides under oxathiin, which are used to manage rust and smart respectively. Then 1967, in this year, dodemorph and tridemorph were introduced. In 1968, we see discovery of benomyl, another systemic fungicide. So we have now every year some developments in new chemicals coming for plant disease management. However, after introduction of systemic fungicides, problems also started coming, like report of fungicide resistance. Within 1 year of introduction of benomyl, fungicide resistance was reported from powdery mildew pathogen, cucurbit powdery mildew pathogen against benomyl. That means the cucurbit powdery mildew pathogen became resistant to the fungicide benomyl. So benomyl was no longer effective to manage cucurbit powdery mildew pathogen. So this case of fungicide resistance, how it develops, how it can be managed, those will be discussed in some other presentations. So that is the drawback of systemic fungicide, because they are single site inhibitors. They affect one point in the target pathogen, metabolic pathway or protein synthesis or cell wall synthesis. So they target only one site, even mitochondrial electron transport chain. So because of this single site inhibition property, systemic fungicides can easily be overcome by pathogens, fungal pathogens. Then in 1970, thiophanates were introduced. Triazoles were introduced as fungicide in 1973. Triazoles, there are many which have been developed. There are many compounds of triazoles which are very effective as fungicide. All of them are systemic in nature. In 1974, the systemic fungicides like prothiocarb, propamocarb, which are effective against Pitheaceous fungi, were also introduced. Now, Pitheaceous fungi, we call it fungi still for easy understanding, but those which are Pitheaceous, they are not true fungi. And it will be explained in some lectures in my channel. You can watch the video. Why oomycetes are not called true fungi. Anyway, Pitheaceous microorganisms were treated as fungi earlier, so this phrase has been used. Then in 1977, introduction of imazalil, effective against benzimidazole resistant strains. Benzimidazole is a systemic fungicide. Now the fungi which became resistant to benzimidazole, they could be controlled by introducing another group of fungicides, imazalil. Then came metalaxyl and furalaxyl. These are also systemic fungicides, very effective against oomycete pathogens. Oomycete pathogens are Pythium, Phytophthora, Peronospora, all downy mildew pathogens, then white rust pathogens, then Pythium, which causes damping of disease in many crops. So these pathogens have been managed by introducing the fungicides like metalaxyl and furalaxyl. Then in 1978, phosphatile aluminium, another very good fungicide against oomycete pathogens, was introduced, phosphatile aluminium. Now since 1970, there has been gradually new fungicides developed: triadimifon, tricyclazole, carbendazim, propiconazole, hexaconazole, myclobutanil, tebuconazole. So all these three here, those were introduced in 1986. Tetraconazole, epoxyconazole, prothioconazole, those were introduced. And then azoxystrobin was introduced in 1992. Trifloxystrobin, fluoxastrobin, those were introduced. These azoxystrobin, trifloxystrobin, and fluoxastrobin, they are from strobilurin fungicides. Now, strobilurin fungicides were basically obtained from a fungus, a mushroom, Strobilurus. Now, this mushroom produces these compounds. When it was discovered, then those compounds were modified, synthesized industrially, and then produced in large scale, and they were used as fungicide. These are very good fungicides. These are broad spectrum systemic fungicides. We'll talk about these strobilurin fungicides in detail in some other lectures. Now, we'll see a brief history of fungicide uses in India. Well, some of them have been mentioned in the earlier slides, but still we'll see here chronologically how fungicides have been used in India. There are many reports, but only some important ones have been presented here. In 1885, 19th century, Ozen first reported copper sulfate, use of copper sulfate in India. In fact, he tried copper sulfate in India to control sorghum smut. In 1904, Lawrence first time used Bordeaux mixture to manage leaf spot of groundnut. In 1909, McRae reported control of blister blight of tea using Bordeaux mixture in Darjeeling. Darjeeling is a place which is famous for tea. So blister blight of tea was managed by using Bordeaux mixture. In 1914, Burns used mercury chloride for the first time in India to treat sweet potato to prevent from rhizoctonia diseases. Hilson also used organomercurials for the first time in India to control sorghum smut. Seed treatment with organomercurials was done to manage brown spot disease of rice. It was done in the year 1929 by Uppal. Brown spot of rice is one disease which is responsible for causing Bengal famine, the Great Bengal Famine of 1942. This brown spot disease of rice was attempted to be managed by organomercurials, seed treatment with organomercurials. Then in 1930, Narasimhan recommended linseed oil in Bordeaux mixture for more coverage and tenacity. Usually, Bordeaux mixture has more tenacity. To further increase the coverage and tenacity, linseed oil was recommended by Narasimhan. Dastur reported the control of foot rot of betel vine with Bordeaux mixture. Then control of powdery mildew of vines using sulfur dust was reported in the year 1931. Sulfur dust is still effective against powdery mildew pathogens. We'll talk about sulfur fungicides in some other presentations. Now these sulfur fungicides are very effective. Elemental sulfur is very effective as dust formulation against powdery mildew. Effectiveness of sulfur dust depends on the particle size. Lesser the size of the particle, more effective it is. And sulfur dust here is a contact fungicide, and there are multiple theories and hypotheses about how these sulfur fungicides, sulfur dust, prevent pathogens like powdery mildew. Then Dastur in 1934 recommended controlling anthracnose disease of cotton by seed dressing with organomercurials. Then control of citrus canker by Bordeaux mixture was also reported by Dastur in 1935. As we said earlier, Bordeaux mixture is a contact fungicide. It is a mixture of copper sulfate, lime, and water. It is not only a fungicide, it is also a bactericide. Many copper compounds are effective as bactericide, like copper sulfate, copper hydroxide, copper oxychloride. Then in 1959, Gruel and Dhorambir first reported control of stem and leaf rust of wheat using nabam and zinc sulfate together. Nabam is also another sulfur fungicide. We'll discuss about this in other presentations. In 1964, Thirumalachar developed one antifungal antibiotic, that is oreofungin. In 1969, Chatrath et al. Reported control of loose smut of wheat. Loose smut of wheat which is caused by Ustilago triticii. It was controlled by using systemic fungicide carboxin. Carboxin, which is the first discovered systemic fungicide in 1966. So immediately after discovery, we see the use of the fungicide in India in the year 1969. Then based on our convenience, we can group the fungicides into first generation fungicides, second generation fungicides, third generation, and new generation fungicides. Certainly, the fungicides which were developed earlier, they were all contact fungicides. We got systemic fungicide in 1966. So this period between 1885 to 1965, spanning over 80 years, it is an era of contact fungicides. Then in 1966 we got systemic fungicide. So 66 to 76, this period is a second generation fungicide. We got many systemic fungicides which are site specific inhibitors. In third generation fungicides, we have many systemic site-specific fungicides which are also broad spectrum. We have got fungicides effective against oomycetes like metalaxyl. Then we have many other fungicides like phenylamide, triazoles, phenylcarbamates. All have been grouped under third generation fungicides. Now, whatever we are having, they are all new generation fungicides, novel fungicides. They have site specific actions. We call them new generation fungicides because we know the fallout of excessive use of fungicides. We know how bad a fungicide chemical can be for the environment. Now we are aware of the environmental consequences of use of fungicides. So our aim is to now develop ecologically safer, broad spectrum fungicides which are required in lower dose and the fungicides which have low toxicity to plant and human. So these are generations of fungicides. So with this, the lecture ends here. If you have any query about this lecture, you can write to me in the comment box. Thank you very much.